Hello everybody, and thanks for coming to this very last talk of the conference. I am Javier Puy, I'm postdoc in Trinity College Dublin, and I will talk you about a new research that recently came out in New Phytologist about transgenerational plasticity triggered by plant competition and how this might affect adaptability and decomposition of the offspring. During this conference, we have seen that individuals, although they're from the same species, although they're from the same population, they are pretty variable in their phenotype. These phenotypic differences might stem from a genetic variation, so some differences in the DNA sequence, but also we have seen from a Thursday afternoon session that it might stem from some epigenetic variation. And as you remember, DNA methylation was one of the most understood mechanisms of epigenetic variation. But not only this, phenotype is also modulated by the environment, because the environment might affect directly how is the phenotypic variation, but also it's interesting because environment might induce changes in the epigenetic mechanism, so and at the same time might affect the phenotype of the individuals. What I'm interested in is in this part of the phenotype. This part of the phenotype, generally called phenotypic plasticity. So how the environment affects the phenotype of the individuals. And it's interesting because this plasticity might occur during the life cycle of a plant or of an individual, the generally called within generation plasticity, but also it might be heritable. So meaning that the environment that the parents lived also affect the offspring phenotype. And in this case, environmentally induced heritable epigenetic modifications might be one of the factors that contribute to this transgenerational plasticity. And why do I say that phenotypic plasticity is ecologically and evolutionarily important? Well, because this phenotypic plasticity affects back the ecological interaction and the environment. For example, it might increase or decrease the success of the offspring. So for example, promoting adaptation. Just think of this example of herbivory triggered plasticity and how this plasticity, this modification, phenotypic modifications might affect back the herbivory, right? And well, this adaptation might affect how is the coexistence and how the community is assembled, but at last it can determine how is the how are the ecosystem processes and functioning. Just think of how these epimutations in Linaria vulgaris might affect the pollination. So basically, this is the frame where our research is located, and we were more specifically interested on how plant biotic interactions are triggering phenotypic plasticity and transgenerational effects. So also checking this heritable phenotypic plasticity. So also, we were very interested on checking whether epigenetic mechanisms are the ones controlling these transgenerational effects. And also, we wanted to test all the consequences that transgenerational effects might have. For example, like promoting adaptation or affecting population and ecosystem functioning and processes. In order to do so, we performed 4 coordinated experiments that all of them have in common that we use one species, this Taraxacum brevicorniculatum, as model species. This species is a bit different because it's an apomictic species, so it means that it makes clones of itself. So offspring is genetically identical than the mothers. We did this in order to reduce the genetic variability as much as possible. Well, first of all, we needed to run a parental generation. In this case, we took our model species and we grow it alone or in competition with a combination of different species. These different species differ in their competitive ability. So in some combinations, our model species was suffering more competition, and in some other competition, our model species was suffering less competition. Already, we found in this parental generation that generally stronger competition trigger plastic modifications towards a more conservative phenotype. For example, they were having higher literary matter content, so denser leaves and coriaceous leaves, and also they were allocating more biomass into the roots, so higher root mass factor. When I refer to a conservative phenotype, means that refers to the resource use and exploitation strategy of the plant, also known as the plant economic spectrum, and is associated with longer lifespan of the individual and better nutrient use conservation. And then we ran some experiments with the offspring. So this first one, we took seeds from this parental generation and we grow all of them in a common environment. What we see from here is that individuals coming from a stronger parental environment, like a more competitive parental environment, they were germinating faster, but also they were growing faster and they were having some more conservative traits as we saw before. Interestingly, when we demethylate the individuals, so for the ones that you don't know what is the methylation, basically we remove the ability of the plants to change their DNA methylation, that like this epigenetic mechanism. So basically, when we demethylate the plants, we stop seeing this pattern. It was not significant anymore. So basically, we erase the memory of this parental environment because all of them were growing at the same rate and they were having similar traits. This gives us the idea that these transgenerational effects were epigenetically controlled. Also, we ran another experiment with the offspring. In this case, we took seeds from the parental generation and we put them in the same competitive environment or in the different combinations. And what we see from this experiment is that individuals, of course, they were responding to the ongoing competitive gradient. So with stronger competition, they were having for example, more conservative traits, but not only this. If the individuals were coming from a stronger parental competition and competitive environment, they were having even more conservative traits, a phenotype more conservative. And one example of this is that is very interesting is the carbohydrates storage in the roots. Basically, individuals, if they were not coming from a very competitive environment, they were just responding to the ongoing competition gradient, so always investing to more carbohydrates when they were under strong competition. But it's funny because individuals that they had this parental competitive background, they were always investing in having more carbohydrate storage in the roots. And then we did a third experiment. As you remember, I told you that we were interested in whether the transgenerational effects were triggering some effects on the ecosystem processes like decomposition. So we did a decomposition experiment. We took leaves from the previous experiment generation, and we performed a decomposition experiment with the leaves. And what we see from here is that in leaves from individuals that were ongoing stronger competition, they were decomposing slower, but also if they were coming from a stronger parental competitive environment, they were also decomposing slower. And this is not just by magic. Of course, these decomposition rates were associated with leaf traits, for example, like leaf chemical traits. And this for us is very interesting because more like conservative phenotypes are the ones that are having slower decomposition rates. But this slower decomposition benefit individuals with conservative phenotype because they have slower resource uptake also. So this led me to the conclusions. We have seen that competitive interactions, competitions, is triggering phenotypic changes, not only within the life, but also heritable ones, these transgenerational effects. We saw that these transgenerational effects were epigenetically controlled, and DNA methylation was one of them. We saw that stronger parental competition is triggering some more competitive phenotypes because they germinate faster, they grow faster, and they have this conservative phenotype. Also, we saw that there is a concordant within and across generational plasticity, and this is interesting because if the environment is kept like this, it might promote some rapid evolution and rapid adaptation. Also, we have seen that plasticity affect ecosystem processes like decomposition. And this is interesting because it allows organisms to shape the environment that they grow in. And at the end, we can say that the transgenerational effects that we saw, they are promoting adaptation, not only via inducing some changes in the phenotypic plasticity, but also promoting some positive plant-soil interactions. If you are more interested about this research, just please check our paper in New Phytologist published last March, or in my Twitter, you might find it. And of course, thanks to everybody involved in this research, and thanks to you for your attention. Thank you very much.